Very good afternoon to everyone, first of all. I will try to be as brief as possible. But today's talk, the subject that I wanted to talk about is specifically one, I hope I'm audible. I'm not sure if the mic is, okay. So it's specifically one fossil from the Manjra Valley site complex in Maharashtra, and the process we use to identify it and why. Like, why is this particular fossil important in the paleontology and paleoecological reconstruction? Acha. Now, tentatively, of course, we identified this specimen as belonging to the Indian tiger, Panthera tigris, identified by Linnaeus in 1758. Now, a few things that are important about the tiger, and especially when we talk about the paleontology of the Manjra Valley, is that first of all, it is one of the largest, it is the largest extant big cat in the world. In India in general, within its habitat, it is the apex predator, and why, particularly its role as an apex predator is very important when we want to study paleoecology. Okay. Now, as with many other species, many other groups of animals, also fossil finds, even though it is a very important animal within the ecology, fossil finds are extremely rare, and we'll go into why also. But before that, I wanted to talk a little bit about why apex predators are important in the study of ecology, both past and present. So, in fact, there's something ma'am also spoke about today, that predator species in general, in absolute numbers, they are very less compared to prey species, right? Which is why we have the triangular food chain or food web. Larger groups of animals, then smaller groups of animals. But the reason why apex predators, particularly the animals at the top of this food chain, are important is that through their existence and their behavior, they affect the environment and the lives of the other animals in many different ways. So just like rodents, we say rodents are environmental or ecological indicators because they represent proof of rodents at a site, whether a modern site or a fossil site, is taken as an indicator of environmental stability for a number of reasons. Particularly because small, short-lived animals are very sensitive to environmental change, and their appearance, extinction, those patterns often correspond with larger climatic and environmental changes. In the same way, but in a reverse, actually kind of in a reverse manner, the presence of predators indicates a healthy enough ecosystem to support them. So what we call this is a top-down control method. So a small population of predators exerts pressure on the biomass of a larger group of prey animals, which in turn exert pressure on the producer species, the plant species. So in a way, the absence and presence, the movement of predators can have a lot of different implications and effects on an environment. And in paleontology, when we reconstruct the environment, when we reconstruct what we call paleoecology, it is also important to understand aspects like this. Now, the problem with studying predators in paleontology is even today, even with the depletion of animal populations in the last 100 years, we still have a huge number of apex predator species in the subcontinent. Okay, there's the tigers, leopards, lions, bears, snow leopards, clouded leopards. But when it comes to fossil finds, we end up with something like this. There are many reasons for this. Now, there are two problems here. First of all is the scanty fossil evidence. There are reasons for this. First of all, like again, in actual numbers, predators are small. There are small populations. And even a large population translates into a very small number of fossils. So for a small animal population, the number of fossils will be even less. Additionally, being somewhat gracile animals, by gracile I mean animals with larger, delicate skeletons, many times the preservation is not that good. And there's a problem not just in India but worldwide. There is actually a gap in the Miocene of 100,000 years where there are no fossils of any kind of big cats visible anywhere in the fossil record. But also then identification is a problem because many of these species, they belong to cats, they belong to dogs, and they belong to some ursine species. And the issue happens with identification is that many of these animals have skeletons that are so similar that it becomes difficult to identify them at the species level. But this species level identification is also important because despite being biologically similar, many of these predator groups have very, very different behaviors, very, very different habitats, very, very different dietary patterns. So taking the example of tigers, for example. Now in the Indian subcontinent, currently the tiger is one of the apex predators, the other being the lion, and then of course wolves and other environments. So I won't go into too much detail with this because it's not mainly, I just want to talk about how cats evolved in general. From a common ancestor, we have the Panthera group. I'm just skipping ahead. Panthera group splitting around 10 million years ago. Okay, now animals like the saber-toothed tigers, we don't really, even though there are some fossil evidence of them in the Shivaliks from the Miocene, particularly 2 or 3 species, Meganterion cultridens and Shiva panthera, but they are not very closely related to the big cats that we have today. So the big cats that are existing in the Indian subcontinent today, they belong to the genus Panthera, which is classified mainly on the basis of certain skull adaptations and behavior-wise the ability to roar. Okay, so out of this Panthera genus, it diverged in 3 different directions. Towards Africa, we had the lion, Panthera leo, the leopard, Panthera pardus. In the Americas, towards the New World, we have the American lion, which was Panthera spilea, and then somewhat related to the modern African lion, but of course it does not exist there anymore. And then towards India, we had the migration, the divergence of the tiger, and 2 sister species which are not classified into Panthera but are similar. These 2 are the snow leopard, Uncia, and the clouded leopard, Neophilus. This shows the historical range of the yellow shows the historical range of the tiger, and the green shows its present day range. So as you can see, it has shrunk down a lot. Out of the subspecies of tigers living in the world today, in the 20th and beginning of the 20th century, we had 8 subspecies in total. Out of those, 3 are extinct now, and 1 more exists only in captivity, the South China tiger. Now, a few important notes about the evolution of the tiger in India. First of all, the map, the green areas show its present day range. The blue areas show where its historical range. So historically, actually, the tiger was there almost throughout the subcontinent. Now, in terms of DNA is where things get a little strange. Most of the DNA studies that have been carried out, such as Kitchener and Dugmore in 2006, they claim that the tiger only came into India in the Holocene period. Okay, and furthermore, another study, Luo et al., also kind of reiterates that based on mitochondrial DNA, the presence of the tiger is only 12 to 14,000 years ago. Now, at the same time, they've also looked at the separation of Panthera tigris, the Indian tiger, from Panthera tigris amoyensis, altaica, the Chinese and the Siberian variants. That, on the other hand, shows a very interesting split around the time of 74 to 108,000 years ago. And this also, some people have taken this divergence as evidence of the effect of the tephra eruption. In the volcanic winter that followed the major Toba tephra eruption, there have been certain changes in other animals also. For example, we see that the migration of elephants out of Africa increases, like at least in terms of fossil evidence, there seems to be evidence of a northward migration immediately following the Toba eruption. And similarly, the Indian tiger, Panthera tigris, appears in the fossil record, genetic record after this. Now, as far as fossil finds are concerned, we do have evidence from mitochondrial DNA, but in terms of fossil evidence, again, we have very, very few. And also, these 3, there are the large cats of the Shivaliks which do not fall into this group at all. I've just mentioned them because they are there. Then the 3 so-called tiger appearances in the fossil record of the Pleistocene. We have them in the Kurnool caves, we have them at the Hunski Bajbal Valley, and we have the species, that's the specimen I'll be talking about now. We have that from the Manjra Valley in Maharashtra. Additionally, in Sri Lanka, some tigers have been identified from the Ratnapura and Batadomba Lena caves. Now, again, the problem here is that these have been identified as Panthera species or Panthera species comparable to tiger, but we don't have that exact species identification. And then other carnivores, of course, like canis, some lion. The lion has been found in sites in North India, particularly in Bengal, and leopard also from the Kurnool caves. Now, the problem, one thing to keep in mind here is that compared to tigers, lions have a very different behavior. They have different behavioral traits. They are social animals. They occupy a different environment. Their prey preferences, hunting methods, these are all quite different. So the reason why we are looking to identify, wherever possible, identify predators at the species level is to make sure that when we are reconstructing paleoenvironment, because a large part of paleoenvironmental reconstruction involves looking at the habitats of the animals we find in the fossil record. And for this reason, species level determination, especially of predator species, becomes very important. So as an example, this particular specimen, which was of course, there's also a very rare fossil find because it's one of the few cases where a humerus, a long bone, has been found intact. And that too a long bone of a predator animal, of a large cat. We know that it's a large cat. Now we'll go to the how. Otherwise, if you see, most of the other finds are either phalanges, finger bones, a broken part of a calcaneum, teeth. So these are very small, dense fossils that tend to survive the taphonomic process. Larger, more delicate bones like the humerus generally do not. Now, a few important things about this is that, first of all, this was found completely in situ. It was found in a deposit from which both shells have been dated and actual fossils were also dated last year, which gave us a date of roughly 21,000 to 24,000 before present. So placing it in the upper Paleolithic. Now, that kind of conflicts with the DNA evidence that we have. The empty DNA evidence argues that tigers have only entered in the Holocene. And one of the justifications that these studies give is that tigers are absent in Sri Lanka, and the so-called tiger fossils are actually some other big cats. So again, a lot of this argument comes from inaccurate identification or lack of inaccurate identification at the species level. So with this particular specimen, we had to try and eliminate as much doubt as possible. Some of the characteristics again, it was found intact and taphonomically. So when we say taphonomy in fossils, we look at the preservation and the actions of different movement, transportation, trampling. Now this specimen luckily has very, very little taphonomy. Apart from some slight damage towards the head and a small breakage towards the distal portion, which is, sorry. Yeah, got it, thanks. Okay. So yeah, apart from slight trampling of the head and a small breakage here, this specimen is mostly intact, and more importantly, there is no evidence of any kind of abrasion, weathering. So we know that there's an autochthonous fossil. It is not from a separate environment, it has not been transported or moved. And the first thing that we did is morphometric analysis. Simply, we take measurements of every single skeletal element, every anatomical feature. We look at their placement and compare it with modern examples. So we compared it with both tiger and lion, around 30 individuals from different collections. Most of them came from the Zoological Survey of India in Calcutta. Now, there were again certain problems with this also. So even in this modern reconstruction, the tigers from the Zoological Survey were mostly Bengal tigers, Sundarban tigers. So again, there is a difference in size. Otherwise, we tried to go for as equidistant, you could say, as distant a population as possible. We had some samples from the Borivali National Park, Sanjay Gandhi National Park in Borivali, of the Western Indian tigers. We had one Siberian tiger which was there in their collection. We took some Bengal tigers and we took some lions. The lions again most of them are Indian lions, but 2 individuals of African lions were also compared. These are some of the lions, Panthera leo. And these are some of the tigers. Now, again, these are from the Zoological Survey collection, and you can see that some of these bones are so old that they're still using felis. Felis was in use before 1929. So part of the problem with studying animals like the tiger in paleontology is that there is very, very little access to the modern animals. And for comparison, since it is a Schedule 1 animal under the Wildlife Protection Act, all skeletons are nowadays destroyed. So the only collections we have are from very, very long ago. And sometimes, as with even the Zoological Survey specimens, we do not have exact context for all of them. We know that they were in the collection, added during British times, but beyond that, we don't know. They took basically length, breadth, height, breadth at different points, breadth at the narrowest point, widest point, breadth of the different distal aspects of the bone. And this is the total number of measurements. This is not that important, but mainly I just wanted to say that all the specimens that were considered for the study were adults. There was mostly male, quite a few females as well. The only issue we had were 2 very young animals, both lions, where the bones were incomplete, because in young animals what happens, the ends of the bone tends to fall off. So preliminary what we saw is that the fossil specimen corresponds more with tigers and lions in terms of absolute size. It was much larger than the lion specimens. General measurements were on the longer side compared to lions. There are a few points, however, especially towards the measurements of the head of the fossil, where the fossil specimen turned out to be a little smaller than the modern specimen. But this could also be an effect of actual damage to the fossil, because like I said, the head of the fossil was slightly trampled, so slightly damaged. Now, these are the comparisons with modern tigers. The fossil specimen the Indian tiger, Bengal tiger, and Siberian tiger. So you'll notice that Siberian tiger obviously is larger, because even today the Siberian tiger is much, much larger than the Indian tiger. Similarly, measurements with lions, where of course this fossil specimen in terms of size is much larger. Likewise, we had also looked at leopard, which are not included here. Leopard is far, far smaller. So where our specimen measures about 280 mm in length, the leopards are much, much smaller. They don't compare anywhere nearby. One thing we did with the actual numbers is that we did something called, yes ma'am, we did something called principal component analysis, which is statistical method of sorting multivariate data, where you have a set of, so each of these dots represents one set of measurements, as in one animal. And in principle, when you put a cluster graph on a principal component analysis, it basically shows you how correlated these different data sets are in terms of their similarity. So the tighter the cluster, the more related the animals are. So in lions, we see there is a very vague cluster being formed. And in tigers, there's a slightly more, like it kind of includes. So our specimen, which is the top left one, this one, is kind of, it's quite separate from lions. But what we see in tigers is because there's so much size variation in tigers, the specimen vaguely falls into that group. But by itself, this is not enough to identify it as tiger. So second thing we did is something called morphological observation. So in other words, we look at the shape instead of the size. Okay, again, similar animals. So in general, the shape of the bones are quite similar. But when we look at individual anatomical features, one by one, we see certain differences, certain very distinct differences between lions and tigers. First of all, the shape of the head. Okay, in lions, the head of the humerus is quite large and it extends longer than that of the tiger. The second difference is something called the deltoid tuberosity is much more oblique in tiger. When we look at our specimen, the head does not extend as much as the lion's does. And secondly, the deltoid tuberosity is more oblique. And interestingly enough, again, so lion, tiger, and the fossil specimen. So in lions, the deltoid tuberosity forms a definite point. So there's a much more prominent point, which is, it's a mechanism for muscle attachment. So in tigers, that is much more smooth than it is for lions. And again, in our specimen also, the deltoid tuberosity does not really come to a point in the way it does with lions. Okay, next one. Looking at the distal side of the bones, this is called the olecranon fossa, which is basically around the elbow. So this in lions, the olecranon fossa is much wider. In tigers, it tends to be on the narrower side. Same thing what we see in the specimen is that however, it is important to note that in the fossil specimen, it's slightly wider than the modern tiger, but not as wide as the lion. Okay, similarly, on the medial side of the bone, there is an extension in the medial condyle, which is very pronounced in tiger, not kind of there in lion. And in the fossil specimen, it's there, but again, not as pronounced as the modern tiger. But it is there. It's not completely flat. Next, now, two very important distinctions where these two specimens differed the most is in this thing. First of all, this keel above the medial condyle. In lions, this keel forms a straight line. In tigers, it goes in an arch form. And this is almost exactly the same between these two specimens, if you notice here. This and this. The second thing is something called the supracondylar foramen. In lions, supracondylar foramen is much more circular. It's much wider, more circular. In tigers, it is very narrow. Okay, it's almost an oval, like we see here. So again, the fossil specimen kind of shows morphologically, more so than looking simply at measurements and numbers, the shape variations in the fossil seem to be much closer to tiger than they are to lion. So in this case, combining this with the morphometric features, combining this with the general the location where the fossil was found, which corresponds both geographically to the modern habitat of the tiger and also in terms of environment, where the Manjra Valley has primarily been seen to be a perennial wetland forest compared to the tropical dry deciduous forest that lions generally prefer, and which also corresponds with the range of the lion seen in terms of the fossil finds in Bakura and other places. So we can and the other thing, of course, is that the prey availability in the Manjra Valley, the number of species that have been found, the tiger has a habit of taking much wider range of prey than lions do. Lions generally stick to ungulates. Tigers have a much more, because they're solitary hunters, they have a much more wider range of prey species. And we generally have a good idea of the, the total number of prey at the Manjra Valley, and interestingly, one other site in the Hunski Bajbal Valley, which has also given a wide variety of prey species, and we have also found one presumably tiger fossil there. So, of course, we cannot make any conclusions based on this because it's only one single fossil in each case. So, statistically, it is not viable at all. But at least it's an interesting coincidence that in the southern Deccan, the only site to give any evidence of a large cat and probably of tiger is one where the faunal diversity is much higher compared to the others. So obviously, again, implications. Now, first of all, since we have the AMS dates on fossils directly from the same deposit as this tiger fossil, we know that it is dating to that time bracket of 21,000 to 24,000 years ago, which does not match at all with the mitochondrial DNA analysis. So that's something which has to be revisited. But the problem is, again, that our fossil evidence is very, very limited. So future directions we can take with this is there are some destructive sampling methods which will end up destroying the specimen. But it might, again, there's no guarantee of it, but we might, if we try to sample it for AMS dating, or if we try to take thin sections for histological study, they might give us more evidence to compound what we are looking at in terms of both time and species identification. But then there are some other methods which are non-destructive, which might give us some more data, and that is what we are moving forward to. And of course, the other issue to address would be the very incomplete fossil record. For example, in Pakistan, even though there is evidence of tigers existing in Pakistan, at least until the early part of the 20th century, we don't have any fossil evidence that has been reported as yet. So these are directions to move forward, and hopefully we get a better idea of predator species within the Indian subcontinent. But at least in this case, we can conclude that this particular fossil was of tiger, which brings with it the implications of environment that we are looking for in the Manjra Valley. Thank you.